presentation is about how to access those data that we presented now in praxis. So the first important point to us is the license, okay? So what use can you make of those data? Are you allowed to reuse them? And so we use the very permissive license, the most permissive license, CC0. It means that you are not even asked to cite us when you use our data. So, of course, it's nice if you cite us. We would like you to cite us if you use our data. But the point is that you can reuse our data in any of your tools, and you don't have the problem of license tag that you need to cite the source of your data, and the source is supposed to cite the source and the source and the source. And you might end up citing a dozen of resources because it's stacked on top of each other. So you use Bgee, you can do whatever you want with those data, okay? In any way possible. You don't have to notify us, you don't have to ask us, you do anything you want. So to obtain Bgee data, you have several methods. One of them is through download files. So you can really import the files on your computer and do whatever you want from them. So there are two levels of information that Bgee provides. So first, the expression calls I just presented before. So when we produce a p-value to say whether the gene is actively expressed or not, then we provide this information per species, and you can retrieve only the information per organ. So for each gene in each organ, you will get a p-value of whether the gene is actively expressed or not. And you have like two flavors, either a simple file where you get just this basic information, gene, organ, p-value, basically, or an advanced file where you can retrieve the information per data type with much more information. And then we provide another type of file with all condition parameters, meaning like with developmental stage, sex, strength. So you have way more rows in that file. This is a much larger file, really taking into account all the information we annotate. And again, either a simple version, just the gene and the p-value, or an advanced file with really all the information we have. And the second type of information we provide is the processed expression value. So we retrieve the individual data set and we process them to give gene expression level information in TPN, for let's say. So we provide this information again per species for each data type individually, so that you can really redo your own analysis. You don't have to redo the read mapping, you don't have to retrieve the raw files from SRA, you don't have to figure out which files were wild type healthy, what were the conditions. We provide everything. And for each data type, there are like two files, but I'm going to show that in the interface after. But you have two files, one with the annotations, so that, for instance, you can ask, give me all the data in brain. So you will have the annotation giving you all the library IDs annotated to brain, for instance. And then you get a second file, which is much larger, which is the gene expression matrices, basically. And so this is an example of what you can find. So in the expression code files, yeah, you get the gene ID, then the anotypical entity ID with the name, the stage ID, sex, strain, and then the expression state at the end, whether it's present or not with the quality level. And you got the p-value and the expression score, so that you can know the level of expression of your gene in that integrated way that is comparable between experiment and species. And for the processed expression values, then you have way more information because it's going to be for each gene, in each sample. You're going to find again this annotation that we provide, but then you're going to have access to the read count, the TPM value, the rank that we use to produce the expression score. But again, you will provide the p-value. So the p-value from each sample. So here is the p-value in each sample. And here at this level, it's the FDR corrected p-value coming from all available information. Okay, so in the expression code file, it is the summarized information. In the process expression values, it is the information from each sample. Okay. So yeah, in our web page, on our web interface, you will find a link download files. And if I click on human, for instance, you can see that we provide information for droplet-based RNA-Seq, so 10x. So you get the annotations, you get the UMI counts per cell population. So we pseudo-bulk the information, or you can find the information per cell in H5AD. Okay. And same for full-length smart-seq data. You get the information per library, but we also, for convenience, provide this information at H5AD files as well, okay? So you get annotation over all the data sets, information per cell population pseudo-bulk, and information per cell. And in this H5AD file, for each cell, you will get the annotation of cell type that is standardized along the UMI counts for each gene, okay? So then other ways to retrieve those data. So either you use our web interface and you manually download the file, or you can use our R package called BGDB. So it is available on Bioconductor. So yeah, you can retrieve the documentation here. You get you get the manual so that you really net all the functions that are described. And using this R package, you can automatically ask, okay, give me all the data sets from Bgee in brain in those species. Give me all the expression quantification, and you can reformat this information into a very well-established expression set object. So then you can integrate those data into your downstream analysis yourself, okay? And the advantage will be that everything would have been standardized, annotated, you know what you're going to retrieve. Another way to retrieve the data, it's more advanced, probably it's really for computer scientists, people doing like data science, I would say, it's to use the Sparkle endpoints. So Sparkle endpoints allows to use the structure of the ontologies, and it allows you to do different types of query, like give me all the organs where this gene is expressed, or give me all the genes that are expressed in that organ. And same with all the developmental stages. And so, yeah, you have an interface here on the Bgee website where here in that text area, you can create your Sparkle query and you get a documentation, you get a tutorial. And so we give example of queries. We explain the ontologies behind. So if you are a data scientist needing RDF data, Sparkle data, you can find them at Bgee. Then you get the web interface, which I'm gonna demo a little bit and then propose to you a little exercise to see if you can make sense of those data. So we have an interface allowing you to query all these data. And basically it's organized in, so you get when you click here on raw data. And it's organized in 3 different categories. You can find the experiments, the raw data annotation, so really the samples, or really the gene expression values, okay? So here, for instance, you can see that we have more than 600 bulk RNA-Seq experiments integrated in Bgee and 41 single-cell RNA-Seq integrated in Bgee so far. And when you browse the data, so you get a form so that you can really perform your query. So let's say I want to retrieve all data in human brain, for instance, including substructure. So if it is annotated to cerebellum, since we use ontologies, we know cerebellum is part of brain. So I can really retrieve all the data that are available. And here it's performing the query, and I have only apparently just one experiment that has been performed on human brain data. I see that here you don't have the count. It probably may be slow because maybe a lot of you are doing the same query. And if I go back, so here it's all the experiments we have, and here on the top you have different fields, and the fields here, they show you the unique value over all the results, because here, like you have, for instance, 600 experiments. If you wanted to know which are the analytical entities, it's not convenient. You have to browse all the results. So here in that field, you retrieve really all the analytical entities for which we have data, okay? And so you have these raw data annotations here. So here it's really for each sample. For each sample, you get the annotation of organ, stage, sex, strain, and everything. And if you go to the process expression value, it's going to be per gene. For each gene in each sample, you're going to retrieve the TPM value and everything, okay? And then from this page of raw data, you can select a specific experiment. So for instance, I don't know if I'm here, I'm interested, let's say, in the fly cell atlas, for instance. So I'm gonna search for fly cell atlas here, submit, I find it here, and I can click on that link, and I end up on the page of that specific experiment. So you get a description, the paper, and then you get download files here again. You can download the data for that specific experiment and not download all the data that are available in Bgee, okay? So here, that's gonna be the result pseudo-bulk per cell population in TSV format. And here it's gonna be the result per cell in H580. And you can browse the result again through the web interface for that specific experiment. So here you can retrieve basically all the cell population that were annotated in Bgee from the fly cell atlas. And again, so you see the free text information provided by the authors, and how do we standardize that in Bgee to map that to an ontology term, okay? So I don't know, like, do I have an example that is complicated here? I don't know, that makes sense here, but okay, you can see how we transfer, how do we standardize the information. Okay, so then other ways, like other programmatic way of accessing our data, we do have a JSON API, so it's more for computational biologists. We do have a documentation allowing you to know how to use this API, for instance, and you can run example queries every time. So for instance, if you want to retrieve the raw data annotation, as I just showed you, let's say I want to retrieve single cell data. Oh, wait, I need to do edit at the top. Yeah. So here you can click on try it out and then test everything. So here, this is an identifier of human. Let's say I want to retrieve single cell data for any genes. I don't filter on that. I can filter on the ethnicity. But here I just want to retrieve all data, let's say, okay? So you can query a specific organ, a specific cell type. I just remove everything here, or a specific experiment, okay? So here I'm asking, give me single cell data in human. You press execute, and it's gonna really show you the query, the URL you are supposed to use, and then it show you the result you obtain. So yes, we do have lots of single cell data in human, okay? So this is the JSON answer. So with this interface, you can really see what are the parameters and try them out, okay? So yeah, as a summary, we provide multiple ways of accessing our data. So the simplest one is to go through our website. You get either the download file that I mentioned at the beginning or this more advanced interface where you can really specify the parameters you're interested in. You can also have these parameters. You can also use these parameters through our R package, our BGDB R package. So most of what we showed on the web interface, you can do using our package directly in R, okay? And then you can also use a Sparkle endpoint if you're interested in RDF data. And you can, oh, that was mentioned, you can use our JSON API, and we have this documentation where you can try out different parameters, okay? So I think I'm good at this point. Yeah, so we have time for questions now.
While people think of questions, I realized that we said we don't integrate circular RNA, but I checked and we do. And I put the link in which shows the utility of our tools, our data access tools. I put the link in the answers. So I went to the data access pages that Frédéric just showed. And in the search for the data experiment, I just typed CIRC, circ, and it completed, and I found circular RNA libraries.
Yeah, the only difference for us is that the set of genes we can study is going to be different. So we are not going to produce a p-value for a poly-A gene if it is a protocol targeting only microRNA, let's say, okay? So that's the only difference for us, but we can integrate any protocol, basically. So...
Well, related question just now, are the expression data for link RNA integrated in Bgee?
So we do have some experiments studying them, yes. But it's quite recent that we target these different type of RNA molecules. So it's not as extensive as for poly-A, okay? It's quite recent that you integrate that. So in the future, you should have more. So I don't know exactly how many we have for those data type. That's what I mean, for those RNA molecules. The majority is poly-A for sure, or ribodepleted maybe. Check whether we have other questions in the documents. Yeah, so Mark is showing how you could search for that. So it's not a complete search because it's just based on whether it was mentioned in the name of the experiment. But yeah, since we don't provide an explicit filter on that, you would need to basically download all the data and filter yourself. But it is a really good suggestion that we should add a filter for those different types of RNA molecules. Yeah, then it is depending on the genome annotation. It is true. I mean, we can only quantify expression of genes that are annotated, right?